Last class you have learnt structure of DNA, structure of nucleosome and transforming principle. In this class we will see biochemical nature of transforming principle. DNA is the genetic material and central dogma of molecular biology. In previous class we discussed transforming principle by Griffith experiment. Once again I am repeating the same. First step, a strain injected into mice, then mice gets died. Second step, R strain injected into mice, the mice alive. Third steps, mice injected by heat killed a strain, then the mice alive. Fourth step, heat killed a strain and R strain injected into mice, the mice gets died. Here a strain transferred material to R strain. Because of this only R strain become virulent. In this experiment, scientist was not explained biochemical nature of genetic material. After him, three scientists, they were Oswald, Pauling and McLean, worked on his experiment. They purified protein, DNA, RNA, etc. from the heat-killed strain bacterium. They discovered DNA alone transferred from A strain to R strain. They also discovered digesting enzymes such as proteases and RNases did not affect transformation. DNA digesting enzyme inhibited transformation. This indicated that DNA caused transformation because DNAs digested the DNA. Hence, transformation did not occur from R strain to A strain. Finally, they concluded that DNA is the hereditary material. But still all the biologists are not convinced. Let's say the DNA is the genetic material. This was proved experimentally by Alfred Hesse and Martha Sayce. They used bacteriophages. Bacteriophage means virus which attack bacteria. Already you know virus has only one genetic material, either RNA or DNA, and surrounded by protein coat. Here bacteriophage contains DNA as a genetic material. These are grown on a medium. Some are grown which contains radioactive phosphorus and some others on sulfur containing medium. Here two different isotopes are used. One is 32 phosphorus isotope in radioactive phosphorus, and while 35 isotopes of sulfur was used as radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contained radioactive DNA but not radioactive protein because DNA contains phosphorus but protein is not having phosphorus. They observed viruses grown on radioactive sulfur contained radioactive protein but not radioactive DNA because DNA does not contain sulfur. These two phages preparations were allowed to infect the culture of E. coli. As the infection proceeded, viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Bacteria which were infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with viruses that had radioactive proteins were not radioactive. This indicated that the proteins did not enter the bacterium from viruses. This proved that DNA is a genetic material that is passed from virus to bacteria. Let's see the central dogma of molecular biology. This was proposed by scientist Crick. It states that the DNA is converted into RNA and the RNA is converted into proteins. Already you know, in some viruses have RNA act as a genetic material. Here, the flow of information is in reverse transcription. That is from RNA to DNA. This process is reverse transcription. Transcription means a gene's DNA sequence is copied to make an RNA molecule. In this process, enzyme used as reverse transcriptase. Here, translation means the process in which ribosomes in the cytoplasm synthesize proteins after the process of transcription of DNA to RNA. Transcription and translation process we will discuss briefly in the next class.